Thank you, Mike, for giving us the opportunity to present part of our joint work. This is joint work with Javier Cervera, Michael Levin, and myself, Salvador Mafe. Javier and me are at the University of Valencia, which is quite old university. It was established more than 5 centuries ago, and Santiago Ramón y Cajal was catedrático de anatomía for a certain period at the university. This is the structure of the talk that we will give. Some material is just to give some scope out of bioelectricity, but the core of the presentation are the points 4 and 5. First of all, I would like to tell you the part of science and technology where Javier and me come from. We have been working with biomimetic nanopores that are fabricated at the Linear Accelerator Unilag at Germany by irradiation with heavy ions of polymer foils. Interesting thing with these pores is that in principle you can attach some chains to the pore surface. For instance, in this case we have attached amino acid chains. With these chains we can control the charge in the pore, and by changing the pH of the solution and the ionic concentration. That means that we are in principle able to have a full description of the system concerning, for instance, current voltage curve. We are getting different rectifying currents, as you can see here. The pores can be used for implementing logical functions, for instance, controlling the current and the voltage with the nanopore. But importantly, the pores work also as rectifying neuromorphic memristors. In this case, for instance, you can see a clear conductance potentiation following voltage pulses. You can also see that the current voltage curve is very dependent on the frequency of the external signal. And we try to do both experiment and theory. These are model results that can be compared with the experiments concerning the frequency and the amplitude of the external signal, and using different salts at different ionic concentrations. It is also possible to implement logic functions not only on the base of this current voltage curve, but also using the conductance potentiation of the pores, which allows a full set of logical responses. It is important to say that we can do signal processing and logical functions not only from the Boolean point of view, but also we can implement reversible logical functions but using arrangements of nanopores. In this reversible function, we can identify the different inputs on the basis of the different outputs, as you can see here. More recently, we have moved to the case of neuromorphic reservoir computing, and in this case we have tried to map a set, in this case a set of 10 digits, by using the conductance response or only of only 5 pores. We send voltage pulses to each pore, and then we try to reproduce in the output, which is the digit we have. We have made experiments not only with ideal digits, but also with corrupted digits. And after a few training epochs, you can get quite good accuracy with these pores. If you change the biomimetic artificial pores and use instead ion channels, you can also study the electrical properties of the ion channel. In this case, we have inserted the outer membrane poring protein of Escherichia coli bacteria in an artificial lipid bilayer. Important difference here is that the currents to be controlled are in the picoampere regime instead of the nanoampere regime, which is typical of the artificial nanopores. Moving to the case of biological patterns, it is quite surprising for an applied physicist like Javier and me that cells can make decisions with only local information. I mean, it's a sort of interplay between short-term and long-term collective persistence. Short-term in the sense that some individual variability is needed for system exploration, for trying to control and trying to understand environmental changes. But these short-term changes should be combined with long-term collective persistence in tissues and organs, for instance. This is quite surprising for a physicist. One possibility to try to simulate this behavior is to use robots, ensembles of robots in this case. Still another possibility is to use some engineering of cell-cell interactions to drive multicellular self-organization. And it's even more interesting to consider agency materials that can show some agent properties and are able to solve problems in well-defined problem spaces, ideally in the case of anatomical morphospaces. But if we go to the case typical in biochemistry, is two schemes dominate or seem to dominate the field, which are the Turing reaction diffusion system based on activators and inhibitors, and the case of the Wolper's positional information concept, which uses one or more morphogene gradients that allow multiple phase decisions for the cell from single or different combinations of molecular gradients. More interesting are the recent agent-based models because they incorporate features from both theories. So, considering this traditional view in the field, we move following Levin's group experiments from the case of the biochemical gradient to the case of biofield gradient. This biofield can be a biomechanical field, which has been explored in the literature, but more interestingly, from our point of view, or at least complementary from our point of view, is the case of the bioelectrical field. In the bioelectrical field, usually gradients of electric potential substitute or complement gradients of concentrations, in this case, morphogens concentration. This is also interesting in the case or in the related case of bioelectrical and biochemical coupling in bacterial communities. In bacterial communities, this work is especially interesting to us in the sense that it's able to couple together chemical gradients and electrical potential gradients. So in this case, it's also very interesting to our field. Moving to the case of bioelectricity, in the last years, and especially in Levin's group, a new single-cell language, which can be complementary to the DNA language, has been put forward, and is the language of bioelectricity based on modern molecular biology, based on proteins, which are ion channels in the case of single-cell case, or intercellular junctions in the case of aggregates of ensembles of multiple cells working together. This adds an interesting computational and cognitive layer to specify multicellular coordination. We will explore here a bioelectrical language which is based on two assumptions. First assumption, the hardware in the computational language is based on ion channels and gap junctions that join two neighboring cells, and especially the cell membrane potential that is generated by ionic concentration gradients and by the conductances of the ion channels, because it is the pattern of cell membrane potentials in a multicellular system that contributes to the coordination of this ensemble. So we move to give a description, but before we have to say that bioelectricity is multi-scale in nature, in the sense that you can find regionalization not only at the level of the ensemble aggregate, but even at the subcellular level. In this case, we have that, for instance, mitochondria have a strong potential difference between the inside and the outside of mitochondria. And this is also the case of lysosomes, for instance, or Golgi or other organelles in the cell. If we move to the single cell case, again, we find cell membrane potential difference, which are significant. And for instance, clear polarized and depolarized voltage potential windows. Usually, differentiated cells are characterized by polarized states in the sense that they tend to show high absolute values of the negative cell membrane potential, while on the contrary, proliferative cells seems to show depolarized values for the cell potential, which here are low absolute values of the potential difference between the cell inside and outside. However, here we are interested not in single cell or subcellular potentials, but in multicellular patterns and collective mechanisms involving cell potential. To this end, we should try first a relatively simple conceptual scheme. We physicists are usually contaminated by simplicity because we are used to work with systems where the degrees of freedom is limited in number. And so we concentrate on only two generic families of ion channels. For instance, a family of inward rectifying uh, channel and another family of outward rectifying channels. We have some experimental excuse to make this first simplification that concerns only voltage-sensitive channels, in the sense that, for instance, homeostatic channel regulation in neurons is based only on a relatively small number of channels than attempt to polarize or depolarize the cell. This is also typical of the cell cycle. In the cell cycle, and the transitions from the polarized cell state to the depolarized cell state, we see that along the cycle and along these transitions, a relatively small number of channels, of voltage channels, are in this case controlling the changes of the cell potential along the cell cycle. If we move to the case of electrogenetic circuits, the design of electrogenetic circuits usually involve only a limited number of counteracting channels. For instance, in this case, transcriptional control of mammalian cells is attempted on the basis of only two channels, a calcium channel and a potassium channel that control the calcium influx following depolarization and repolarization phenomena. Still another example concerning bionic differentiation. Here, together with receptor ligand transporters, two channels, potassium channel and calcium channel, compete to establish the inside cell levels of calcium that downstream control the timing of pluripotency genes. Another example is excitation transcription in synaptic plasticity, where calcium channels are also important, and in particular, the calcium entry to ion channels activates calcium-sensitive transcriptional signaling cascades, always downstream. And in these cases, we usually observe bistable cell membrane potentials. Here there is a clear bistability between a depolarized value of the cell potential and a polarized value of cell potential. And remarkably, there is a clear correlation between bistable cell membrane potentials and bistable calcium concentrations. Remember here that calcium is a second messenger, it's a signaling ion, And this biostability, and in particular this correlation of the biostability between cell potential and calcium concentration should be significant in transcription, for instance. Also, other examples concerning frogs, for instance, as model animals, show that potential windows are important for processes and for downstream developmental phenomena. And again, these cell potential windows seems to be correlated with calcium signals in this case. Plants are also interested from the point of view that you can find multi-scale phenomena where oscillatory and stable potentials can be defined. And this multi-scale phenomena is multi-scale not only in terms of time, but also in terms of spatial scales. So again, we have another example where a transcription can be coupled to cell membrane potentials in a different part of nature. Summing up, many cases tend to suggest that bioelectricity can be coupled with protein expression, and suggest also that a relatively small number of counteracting voltage gate channels trying to fix polarized and depolarized states in the cell seem to be central to the problem. Although we should admit that in specific cases, other ligand-gated channels can also influence these complex downstream processes. Moving to the case of multi-scale bioelectricity, we will consider especially the excellent experimental work by Levin's group concerning model animals like frogs, for instance, or flatworms. This work is quite well known in the group, so we will not insist in the group. We will insist only in some characteristics. First characteristics, the coexistence of depolarized and polarized regions, which are separated by relatively sharp interfaces. Other special characteristic, the possibility of pattern completion by bioelectrical modules. These modules can be externally modified by actions on ion channels and junctions. Another possibility from the point of view of experiments to correct for degraded patterns by using, for instance, the upregulation or the downregulation of specific ion channels and gap junctions. So when Javier and me attempted to first put forward a model with your cooperation, we thought that the model should explain at least qualitatively these experimental facts. Crucial points for a simple bioelectrical model. First, ion channels, which are voltage sensitive, tend to operate over relatively wide voltage windows. This is good from the point of view of getting a rich pattern of responses, but is also bad in the sense that some context-sensitive scenario is to be anticipated because of the wide voltage window. A given ion channel can polarize or depolarize the cell depending on the particular cell state and environmental condition. First, there should be some reason to consider a small number of channels, and especially voltage-sensitive channels, because neurons, in principle, come from non-excitable cells, and neurons are, in the case of the action potential, be controlled by a relative small number, in this case, sodium and potassium channels. Still, another question to be taken into account is that it is possible in artificial biosynthetic excitable tissues to reproduce biostability phenomena and to reproduce oscillatory behavior by using only a limited number, a small number of ion channels and gap junctions, which means that in principle, with a limited number of ion channels and junctions, very interesting biological-like phenomena can be described. So let's move to our model and consider first the case of the inward rectifying channels. To this end, we have considered generic pole conductance families, which represents a diverse family of channels that act to polarize, act to fix the polarized cell state. These channels can be relatively easily described by current voltage curves, which depends on the equilibrium potential. For instance, an equilibrium, let's say potassium potential, and which is very important, voltage-dependent conductance. The channel conductance should be voltage-dependent to reproduce the experimental results in this case. Important thing when we try to understand the current voltage curve is that small deviations from the equilibrium potential can be compensated. I mean, there is a homeostatic voltage region around this polarized cell potential where small deviations can be compensated. However, if the deviation is high enough, if the cell suffers a strong depolarization and move to low in absolute value cell potentials, channel closes. And because channel closes, bioelectrical regionalization should be possible. This is an interesting property of the model we are trying to put forward. Now we move to the case of the outward rectifying channels. In the case of the outward rectifying channels, we have a multitude of experimental curves concerning, for instance, this calcium channel or this potassium channel. So we follow a generic conductance simulating a diverse family of channels trying to depolarize the cell. This is the current voltage curve we are using, which has some difference in the sign of the exponential, and this difference in the sign of the exponential gives the different behavior that can be observed in this slide. In this slide, we see that in the case of the channel trying to fix the depolarizing cell state, there is also a homeostatic voltage region, so that the small deviations from a cell which is depolarized can be corrected, but if there is a strong repolarization, the repolarization could not be corrected because again the channel closes. Channel closes from the transition or by the transition from the depolarized to the polarized cell state, but this channel, this counteracting channel, can also close when the transition is following the opposite behavior from the polarized to the depolarized cell state. Again, this context-sensitive property should be okay, should be interesting for bioelectrical regionalization. Next slide shows the action of the two channels. Summing up the two channels, we get a total current, which for a certain value of the cell parameters can show biostability in the sense that there is an unstable point which moves to the polarized cell state or the depolarized cell state depending on the environmental conditions and the particular initial cell state. This is clearly seen in this graph where there is moving from an unstable point either to the depolarized cell state or the polarized cell state. This moving is highly context sensitive, as you can see. Which is important here is that when you have a perturbation or disruptive event, if the perturbation is weak, the cell potential, in this case the depolarized cell potential, does not change. After a short time of the perturbation acting here, the initial depolarized cell state is recovered. However, when the perturbation in the channel conductance is strong enough, the change from the depolarized to the polarized cell state is permanent. That means that in principle, transient limited in time actions can change the bioelectric cell state if these actions are strong enough. From the point of view of mathematical physics, this can be understood simply by plotting the change of the cell potential with time and trying to obtain these changes for different ratios, for different relative value between the polarizing channel conductance and the depolarizing channel conductance. So having described how ion channel proteins can contribute to the cell membrane potential, we address now a central part in the model, which is how the feedback between bioelectrical and transcription can regulate the protein expression. To this end, we follow these experimental results based on only two counteracting channels. Key hypothesis here, key assumption here is the existence of a local regulatory agent whose concentration is modulated by the cell potential. Because some channel can be open state, can be closed state. This is the key of the model in the sense that if the inside local concentration of this agent depends on the cell potential, because the entry depends on the voltage gated channel, then the cell potential should be able to regulate transcription, because downstream it is this signaling ion or this neuromodulator inside cell levels which influences transcription. So this feedback is clearly seen in the next transparency, where we have connected the bioelectrical layer, which is based on the ion channel dynamics, with the transcriptional layer, which is based on the ion channel expression following kinetic rate equations for the mRNA and for the protein. This is the case of the depolarizing channel protein. This is the case of the polarizing channel protein. These kinetic equations for the mRNA and for the protein should be coupled, because eventually they give some concentration for the proteins, and this concentration from the ion channel proteins give the conductance, and this conductance eventually enters in the equations for the bioelectrical activity. So in principle, we have a model which is able to connect, to couple the transcriptional and the bioelectrical layer. And this coupling is seen here in these equations, because the rates for the transcription depend on the signaling ion or neuromodulator concentration inside the cell, and in turn, these concentrations depend on the cell potential. So this is clearly the coupling. Cell potential gives signaling ion or neuromodulator concentration, and this inside concentration influences the rate of transcription. So we have connected the bioelectrical layer with the transcriptional layer thanks to this equation. In the next slide, you can find the meaning, the significance of every parameter of the model, for instance, conductance, concentrations, and rates. And here we have a typical example of the results that can be obtained with this sort of equations, or this sort of systems equations. And this example is interesting because it's quite simple. We have only a signaling ion concentration, which is dictated by the cell potential because of the voltage-gated ion channels that can open or close. And this signaling ion concentration influences the transcription of the depolarizing channel, and influences, in this case positively, upregulating, in this case negatively, downregulating the transcription of the polarizing channel. That means that you can control the cell potential because polarizing channel tends to increase cell potential in absolute value. You can control cell potential because depolarizing channel tends to decrease cell potential in absolute value, and you have then decoupled between the transcriptional layer and the bioelectrical layer. Clearly, this is a highly simplified scheme. The scheme can be extended to other proteins inside the cell as you need in every case. But this simple scheme shows very interesting properties. For instance, it can show oscillatory behavior for the cell potential between the depolarized and polarized state, but it can also lead to a stable depolarized states and stable polarized states for the cell. Everything depends on the ratio of the channel conductances, which, because channels are protein, is connected to the transcriptional layer here. This simple scheme leads to a configurational phase space like this. Remember that we are applied physicists, and applied physicists are extremely happy if we are able to construct, to describe configurational phase spaces. In this phase space, we have a polarized region and the polarized region as a function of the equilibrium polarizing potential, for instance, due to potassium ion. And this is the ratio between the channel conductances. In between the polarized and depolarized region, an oscillatory region can be obtained. It is important to emphasize that if you change the external or the endogenous conditions, you get a change immediately in the context-sensitive bioelectrical genetic coupling, and then you can change the single cell state, moving, for instance, from the polarized to the oscillatory regime or from the oscillatory to the depolarized regime. So in this case, you can have a theoretical phase space that can try to suggest or can try to explain external actions or endogenous processes within the cell. The limitations of this single cell model is that is not only system dependent. I mean, you should change your scheme according to your system. It is also highly context dependent in the sense that small deviations, small changes in the environment, for instance, can lead to different cell potential states. So this should be taken into account when trying to model the system or to act externally on the system. But the important thing is that we have a relatively simple theoretical scheme that considers the cell as a dynamical system, and this dynamical system can show transitions between the depolarized and the polarized cell states. These transitions can be in principle regulated by the ionic concentrations and by the relative values of the polarizing and depolarizing channel. However, it is clear that to try to understand qualitatively bioelectrical experiments with model systems, with model animals, we need to extend the model to the case of intercellular gap junction to consider multicellular ensembles. To this end, we try to understand better, which means to have a bioelectrical regionalization with sharp boundaries between depolarized and polarized cell states. And this points out the need to consider the potential difference between two neighboring cells. These experimental results immediately suggest that a new key parameter is the intercellular potential difference between two neighboring cells. It is not enough to consider only a cell single state. We should consider at least cells that are coupled with junctions between them. To this end, we again go to experimental data and concerning the carrying voltage curves obtained with typical, for instance, connexin proteins, in this case connexin 43 and connexin 40. This current voltage curve immediately leads to experimental belt-shaped conductances, and these belt-shaped conductances are modeled here with a relatively simple equation, which immediately gives current voltage curves for the junctions that reproduce qualitatively the experimental data. Again, the important thing with these theoretical curves is that there is a central region which is homeostatic in nature. I mean, when you have similar polarized or depolarized cell states, the potential difference between the two neighboring cells is zero, and small deviations immediately give intercellular currents that can be compensated. However, and this is very important, the connexins show this negative differential resistance region, which allows closing the gap junction when the deviation from isopotential neighboring cells occurs. This means that in principle we have the needed inhibitory connections that should permit bioelectrical regionalization as shown in developmental experiments and show this contrast dynamic sharpening. So in principle, we have the ingredients from the point of view of single cell parameters and from the point of view of neighboring cells to put forward a model. But before that, it's important to say that in this model, where the cell state is controlled by the voltage-gated ion channels and the multicellular states are controlled by the gap junctions, in this model, the gap junctions can show very different conductance versus transcriptional voltage, in the sense that, for instance, connexin 45 get open over a voltage window which is quite wide, while on the contrary, connexins 45 tend to be sharp, tend to respond sharply, and tend to close rather rapidly. This sensitivity to the transjunction voltage should promote bioelectrical regionalization, in some cases more easily, in other cases not so easily. Just interesting to note, although the problem is highly more complex, that in glioblastoma cancer stem cells, different junctions and different voltage sensitivities seems to influence in multicellular domains. This can be found in this cell reports work, for instance. And so, in principle, this different sensitivity of transjunctional voltage can be important from my experimental point of view. Note here that in this case is very complex, far more complex than our analysis, and clearly other biochemical and biomechanical effects must also be present in this complex process. But the fact is that this voltage sensitivity of the junction seem to play some role, as explained in this previous article by other authors. Additionally, we can work with models with strong and with weak or intermediate, in this case, ion channels, by changing the parameters of the conductance of the gap junction. And moreover, we can also consider inhomogeneous distributions of weak and strong gap junctions. That means that in principle we can recreate a rich network connectivity dynamics as suggested by the variety of bioelectrical patterns that are found experimentally. This is an attractive point of this sort of relatively simple bioelectrical models. So in principle, we can move from single cell phase space. Remember, please, that this was only a single cell phase space. We can move from single phase space to more complicated, in this case, multicellular, or at least couple of cells connected by gap junctions, and we can try to design or to obtain theoretically different regions explaining an isopotential system with, in this case, isopotential system, for instance, two head, a system which has a clear potential difference between cells in the head, cells in the tail, in this case, head-tail geometry. And importantly, relatively simple equations show the possibility of having biostability between these biostable system states. Another thing to be taken into account is that when we connect two neighboring cells with a gap junction, we not only permit a current, which is intercellular in nature, what we are doing crucially is to substitute a single cell state by a cytoplasm connected cell state, which is multicellular in nature. I mean, there is some sense of belonging to the same isopotential region when lots of cells are connected cytoplasmatically, so they share the same cytoplasma in this sense. This means that in principle you can regulate as a whole a multicellular aggregate using this connection between neighboring cytoplasm in the cells. We have put forward this idea considering the case of short non-coding RNA molecules that can be in principle transferred through intercellular gap junction because of the small size. Some short MR, in this case micro RNA molecules, can be transferred through the intercellular junction and then can affect the ion channels. This should give an additional experimental possibility to upregulate or downregulate certain channels. In this case, in this model, we have assumed that this transference can downregulate a given channel so that the other channel can predominate. We have extended the previous model to this case, and we have an equation for the diffusion of the non-coding RNA through the gap junction, which should be solved in the multicellular cell ensemble together with the bioelectrical, in this case, equation. When we solve this, we indeed find that an initially multicellular depolarized memory state can be, because of the transference of the non-coding RNA, be changed to a polarized state, because in this case we are affecting the depolarizing channel. If we are down-regulating the depolarizing channel, it is possible that the polarizing channel eventually dominates, and we can convert an initially multicellular depolarized memory in a final multicellular polarized memory. These simulations are quite interesting because they show another characteristic of the model and can be found in this reference, which is noted here. Experimentally, gap junction assisted is also a case of set of phenomena that can be observed in other biochemically related contexts, for instance, reaction diffusion phenomena and scheme patterns. Also, gap junctions allow many external actions. For instance, octanol blocking can affect the planaria head morphology, and we have also put forward a model in cooperation with Michael Levin, and in this model we consider that it is the morphogene concentration profiles that are able to lead to different head shapes. And when we block the intercellular gap junctions in the model, we immediately change the concentration profiles of the different morphogenes, which leads immediately, at least from a theoretical point of view, to different developmental processes in the head and eventually to different head morphologies. So gap junction in the model offer a lot of possibilities to consider small asymmetries, local disruptive events, changes in environmental conditions, and so on. In other words, we think as physicists that the progress from the individual cell to the multicellularity immediately leads to average biophysical fields which can be exploited in external actions, in development and regeneration, because they allow a high level of abstraction where the number of parameters, the degree of freedom immediately decreases dramatically, and only a relatively small number of parameters characterizing these average biophysical fields need ideally to be considered. So let's go to some examples. First, it is clear that complex phenomena in physics, such as symmetry breaking and shape regulation, can arise in relatively simple systems when you have multiple units interacting in the system. This is a trivial left-right establishment. Another not so trivial is the case of the Rayleigh-Bénard oil molecules. In this case, these Rayleigh-Bénard oil molecules structures are found when you increase a thermal gradient, a temperature difference, over a given threshold. Immediately in a system which is disordered in nature, immediately ordered structures appear, and this nonlinearity may mimic some self-organization in living systems, in complex living systems where many basic units are interacting. The problem here is that in contrast to the universality of phase transitions in thermodynamics and statistical physics, no general theory seems to be able to predict which organization arises far from equilibrium in biological systems, because specific details matter almost always. Here we have two bioelectrical features in mind which should be reproduced by our model. We have insisted on this feature. First feature, we have to be able to explain contrast, dynamic sharpening, and bioelectrical regionalization. We should be able to show that a degraded pattern can be recovered, that corrupted images can be locally corrected by means of changing the cell depolarization or polarization state, or by means of changes in the connectivity. This should be two interesting things that should be explained in our model. To this end, we try to explain first the bioelectrical community effect. This effect is better explained with an example here. Imagine that the cells are disconnected. The cells are initially in a polarized state, but some change, for instance, an external action on a central cell, causes a depolarization. If the cells are not connected, if the number of effective neighbors is zero, nothing is going to try to reestablish the initial situation. Nothing will be able to keep the cell in the initial polarized state. The cell immediately depolarizes with time. However, if we begin to connect this central cell with neighboring cells, when we increase the number of cells, we see that correcting for the depolarization is possible. And in fact, the cell can be repolarized, the central cell can be repolarized after a time that depends on the connectivity and depends also on the degree of the initial depolarization. That means that we have a sort of a bioelectrical buffer effect because of the neighboring cells. More than this, and this could be important, each central cell can measure the number of neighbors not only by biomechanics but also by bioelectrical effects. Bioelectrical effects should be able to tell the central cell how neighbors are effectively connected to this central cell, which can be important, for instance, in developmental processes, this sort of measuring the bioelectrical of neighbors. This can be extended to a bigger cell community, and in this case, we see that when the region that has been depolarized is a relatively small region. Repolarization forced by the neighboring cells external to the central cells is possible. However, when this abnormally depolarized group of cells is high enough in size, the contrary process is possible and the abnormal state, instead of being normalized, causes an extension to the surrounding cells where initially had, as you can see, polarized states. So the intercellular connectivity could normalize small regions initially when the abnormal region is small, but can also spread the abnormal cell state later when this initial region is big enough, is high enough in size. Pre-patterns of different single cell state can be designed and can be put forward here in this model. And also depolarization waves are possible. You can in principle recruit the neighboring cells to a central patch which has been grafted or which has been modified externally. You can recruit in principle the external cells which surrounds this patch if the single cell states verify certain initial conditions. An operational limitation here is that this recruiting is not always possible in the simulations because it depends critically on the relative stability of the initial model polarization and depolarization cell states. But in some cases, the simulation shows that this recruiting is possible. However, the system-dependent behavior is clear. When we made different simulations, we found that at early stages, when the number of abnormal depolarized cells is small, some normalization is possible, but when the connectivity is low or the abnormal cell states are high in number, this normalization is not possible. So this suggests that at initial stages, if you want to suppress some abnormal bioelectrical state, it is better to increase connectivity, but at a later stage, when the region is, the abnormal region is high enough in size, maybe it's better to decrease rather than increase the connectivity because this will decrease the internal cohesion of the abnormal group of cells. You can find many different simulations in the next slide. And these simulations concern different single cell states that allow correction. Other single cell states they do not allow corrections, and size effects are very important. Not only the size, but the patches, the number of patches is important. In general, a small number of patches or small patches are easier to correct than big patches. Some biochemical reminiscence can be found in other systems that can be explored further in other articles that are also given in the reference list. If you go to correction of degraded patterns, which is also very interesting, for instance, in Levin's experiments, Levin's group experiments has found the possibility of in this case, restore corrupted spatial temporal maps. For instance, in these simulations with my Levin, we try to recover the original polarized state of this region in this model aggregate by an external action. For instance, injection of an mRNA that gives the protein corresponding to the polarizing channel. And we indeed find that corrections of degraded patterns are possible under certain conditions, but again, the context-sensitive phenomena arises, and sometimes corrections are possible, and other times corrections are not depending on the single cell states, and importantly, depending on the system connectivity which we have assumed. We made some qualitative simulations cooperation with by Levin and Pi and Dr. Pi. And what we found in these simulations is that in principle, polarization waves are possible to correct degraded electric potential patterns in this case in the head of the frog of the model animal. We simulated the profile of the electric potential in cases where corrections were possible and other cases where corrections were not possible. And again, we found that the size of the correcting patch and the distance of the correcting patch to the zone to be corrected, to the region to be corrected, are important. There are important size and distance effects, showing again the context sensitive characteristic of the model that seems to be also present in the experiment, these context sensitive characteristics. Another possibility which has not been explored by us in the model, except for some trivial case, is the possibility of using charged nanoparticles. Many nanoparticles can be nowadays functionalized with, for instance, carboxylic acid groups or amino modified groups. These groups should be negative or positive charges attached to the nanoparticles depending on the local pH and ionic solution concentration. It could be expected, according to the model, that positively charged nanoparticles will accumulate in the region of the multicellular aggregate, which is characterized by the cell potential polarized state because of the more negative cell membrane potential. On the contrary, these positive nanoparticles will tend to be depleted from this zone, the region of the multicellular state characterized by a depolarized potential. That means that in principle you can use negatively charged, or in this case positively charged nanoparticles to attempt to change cell membrane potentials. The idea could be interesting, but not that experimentally when it is attempted, not only the charge of the nanoparticle, but also the hydrophobic and hydrophilic properties of the nanoparticle surface is important because it can regulate the nanoparticle binding to the cellular membrane in addition to the case of the charge of the nanoparticle. We have also tried to simulate oscillatory multicellular potentials in the sense that it has been observed these sort of potentials also in experiments where, for instance, some regionalization is obtained by grafting a central region. What we have found is that this central region, which has been grafted in a initially polarized complete system, is able to resist normalization for low enough connectivities, but normalization tends to proceed for big connectivity. However, it's very interesting to have a central collective oscillation when the connectivity has intermediate values, as is this case. So again, we try to reproduce a configuration space with polarized and depolarized states for the central region, which has been grafted. And we found again here that by changing the external or the endogenous conditions, for instance, the connectivity of the gap junctions, transitions can be implemented between polarized and depolarized states, and oscillatory behavior can also be established. To analyze better this case, we considered in other simulations the possibility of having two types of oscillatory cells in the ensemble. These cells are characterized by relatively high periods. These cells are characterized by relatively low periods. When we increase the connectivity of the system, it seems that one group of cells dominates. In this case, dominates the low period cell, and this can be observed in the imaginary versus real part of the control complex number zeta that characterizes the oscillatory behavior. a biochemical reminiscence could be here the loss of oscillations and the recover the oscillations when we reaggregate the systems and increase the connectivity of the system. We have also studied more recently with Michael Levin the case of symmetry breaking amplification by transcriptional waves, by the coupling of bioelectrical and transcriptional waves. Which is interesting is that when we define allow polarizing cell state and in this case high polarizing cell state because of symmetry breaking, not only we can have stable, in this case, electrical potential profiles, but we can found the coexistence of phase and antiphase regions, phase and antiphase regions, which maybe could be developmentally relevant from the point of view of bioelectricity. For instance, we can set a cell point 1 here, cell point 2 here, and see what happens at zero connectivity. At zero connectivity, we can only have the trivial two-region encoding where cells in region 1 are all the time polarized, depolarized, and cells in region 2 are all the time polarized. This is a trivial encoding. At high connectivity, which is the contrary, on the contrary, which is the opposite case, in this case, a homogeneous isopotential system is obtained where nothing can be encoded. Nothing can be encoded because the whole system is as isopotential and the same cell state, in this case the polarized state, is common to the two cells. But for intermediate connectivity, you can couple the left and right regions in phase and antiphase oscillations. You can synchronize the system of distant cells, cells 1 and 2, and this is not trivial at all because it seems to show that there is a critical region of connectivities where even phase and antiphase oscillations can be established. To consider the final part of these examples we have explored with the model, we have the case of planaria and regeneration and acquired tolerance to toxic environments. In this case, we consider the exposition of planaria to barium and try to explain the adaptation to toxicity after some time has passed. We assume in the model this simplified scheme that connects the polarizing channel, which is a potassium channel here, a depolarizing channel, for instance, a calcium channel, and a assumed rescue channel that can enter when the polarized channel is blocked by, in this case, by the barium. The blocking of the channel by barium causes a depolarization of the cell so that the single cell potential move from the polarized value to the depolarized value. Experimentally, this seems to the degradation of the head of the animal where this polarizing channel is established. And eventually in the model, some rescue channel appears that is able to recover, to make the transition again to the repolarized state. And the repolarized state is in principle able to reestablish the physiological potential window. This is the single cell model we consider. And in this model, maybe we consider maybe that barium is not common to planaria in the usual environment, but the planaria has a long evolutionary history, and in this evolutionary history, it should be able to face depolarizations regardless of this depolarization being established by barium or biodin blocking agent. So we assume that in principle some additional protein, in this case a rescue, a compensatory channel protein is triggered, is upregulated by the depolarization. We connect all the cells in the model ensemble and we found a relatively interesting result in the sense that these white cells, which are in the model dead cells that eventually die because of the depolarization that takes the cell potential out of the physiological potential window, they die, but they extend to the system. And this extension to the system, when we connect the cells, is quite limited in the sense that the bioelectrical buffer effect can limit the damage here clearly. So it seems that the model predicts not only a single cell state effect, but also a bioelectrical and multicellular effect, which is due to the cell connectivity in this case. This is at least the prediction of the model, which shows the importance of connectivity in model simulations. We should say that other simulations or other systems are possible, in particular the system by Dr. Alexis Pietak and Professor Michael Levin consider other systems which are in many senses more complete than our simulation procedures. We are physicists and we try to concentrate on relatively simple systems compared to the real systems that are explored by this more complete algorithm by Alexis and Michael Levin. Let's finish the talk admitting that some limitations to this sort of equations we have used is that we have ignored so far biomechanical effects. Biomechanical effects are present in many important systems. In particular, mechanoelectroosmotic equilibrium is central to the cell equilibria. Also, the coupling between bioelectrical and biomechanical networks is well known in model animals. For instance, this work is also by Levin's group. Also, cell proliferation and physicochemical properties of lipids suggest some biomechanical field effect transistor, in the sense that first, there is in this particular problem that can be found here, there is first membrane depolarization, bioelectrical effect. This membrane depolarization causes a biomechanical effect with this accumulation here. And this biomechanical effect eventually downstream causes a biochemical effect. So these real problems tends to show, gastrolite is another problem, tend to show that biomechanics can be coupled to bioelectricity in many cases. Also BioBotch, if you want to recreate BioBotch in modeling, biomechanics is essential. And there are some models that try to connect biomechanics with transcription, biomechanical networks with biochemical networks, and also bioelectricity with biomechanics, that show that not only the sizes and the distance, but the shape matters. And this is well known. The biological physics of developing embryo has traditionally put biomechanics in a central role. Again, another limitation of the model we have explained before is biological complexity. When trying to apply our model to real cases, an extension of the model should consider specific ion pumps and ligand gate ions, which we have not considered. But again, we think that the progress from individual cell to multicellularity immediately leads to average fields. And these average fields should be a complementary method to cell level management, because they are characterized by higher levels of abstraction. We are probably contaminated by our applied physicist origin, and in particular, we have been teaching thermodynamics and statistical physics courses for a long time. So we have in mind all the time that units not should be necessarily equal for robust collective responses. We know that diversity can be compensated by connectivity and redundancy mechanisms. We know that low-dimensional average responses tend to demand usually small system resources compared to the exhaustive control of every unit at the single cell level. And we know that intercellular connectivity is very important and should offer new opportunities. It is true that this connectivity gives the system also some vulnerability to external actions, but this vulnerability can be avoided in principle, and this intercellular connectivity offers also positive aspects, for instance, for external control of the system. From a computational point of view, maybe programming at the bottom layer should lead also uncertain consequences on the upper layer. So the model of bioelectricity seems to offer a context-sensitive landscape concerning average multicellular fields, but it is also true that programming at the bottom layer sometimes have uncertain consequences on upper layers. Still another thing we like of these average fields is that in principle they are governed by a relatively small number of parameters, and when you have a relative number of parameters experimentally, you can test reductionist procedures quite easily in model systems, because the number of parameters to be investigated, to be studied is small. Let's finish with some important warning. As what we can observe is that external actions are very context sensitive because of single cell and multicellular feedbacks, even with relatively simple models. That means that in order to implement particular actions on model systems at different levels of biological organization, a detailed knowledge of target and anti-target channels and possible feedback mechanism is required to implement any bioelectrical actions. According to this simple model, there always has to face some risk of unknown unintended events because of this complicated, complex feedback between different levels of description, in this case, bioelectricity and transcription. And this is all. Thank you very much for the talk. It has been a little bit maybe long compared with my initial intention. I have tried to explain everything maybe with some excellently in terms of the details. In any case, you can find more information. You can find even this PDF file in our webpage. We must thank the institutions that has funded our work. We must find also other cooperators, especially with APAI and Alexis Pietak. And these are some recent covers concerning our bioelectrical and our biomimetic nanopores field. Thank you very much for your attention, and sorry for the rather long talk.